Okay, good evening everybody. I think we'll make a start. It's fantastic to see so many people here for this evening's Pewterers lecture. I'm Mike Hanna. It's my privilege to be the director of the Institute, and I particularly want to welcome colleagues from the Worshipful company of Pewterers, including Charles Robinson, the upper warden, and Mike Walliker, the CEO. I'm also very pleased to welcome our Provost, Michael Spence, who's kindly agreed to do the vote of thanks. He was here a minute ago, has he left? He's gone to the back, okay. And also Alan Thompson, the Dean of our faculty, and also Stephanie Scourge, who has two hats, because she's obviously a UCL professor, but also a freeman of the company of Pewterers, and she's the chair of the 500th Trust, which you will hear more about, but the clue is in the number. So the relationship between the Institute and UCL and the Pewterers goes back 50 years, and we greatly value this close relationship, which has enabled us to have 12 Pewterers Fellows. And I'm very pleased to ask Charles to maybe say a few words at this point about the Pewterers, if you're happy to do that.
Thank you very much, and I will keep the words very few and limited. It's a great pleasure to be here today. I can see several members of the Pewterers company, and for those of you looking at young people, you may not be familiar with what the Pewterers. We're a city guild, and we were incorporated 551 years ago. As you may know, pewter used to have lead in it. It hasn't for a long time. And it was discovered many centuries later that lead damaged the brain when consumed. And so on the 500th anniversary of our incorporation in 1974, if my maths is right, we got together with the Institute of Neurology and formed a charity to fund a fellow here. It's been a terrific relationship. Our current fellow is Sahil Patel. We have a trustee, Stephanie Scourge, who's a member of the company and was introduced, I believe, Stephanie, through your role as the fellow. So thank you very much for having us today, and a special thanks to our speaker, Sarah Tabrizi. We very much look forward to hearing from you. If I could just add one thing, I am a stand-in for our master, Isabel Martinson, who sadly can't be here today because of family circumstances, but she does. She's very sorry not to be here.
Okay, so in addition to funding what have been 12 fantastic fellows over the years who've done great science in a range of brain diseases, part of our relationship has been to hold this Pewterers lecture every 2 to 3 years, which has been happening since 1974. And this has been delivered by a series of outstanding scientists in different areas of brain disease, and tonight is no exception. So I'm very pleased to welcome Professor Sarah Tabrizi to give the 2025 Pewterers lecture. For those of you who don't know Sarah, there must be very few of you. So Sarah is director of the UCL HD Centre, which she co-established with Jill Bates. She's the joint head of the Department of Neurodegenerative Diseases. She's an honorary consultant neurologist here in the hospital with a major clinical practice in Huntington's disease, looking after patients and families. She leads an internationally recognized basic science and translational research program in Huntington's. She's led multiple first-in-man clinical trials, including gene therapies and Huntington's disease, and is absolutely at the forefront of the field, and we're all extremely proud of the work that she and her team does. She's received many international awards, but perhaps most notably in 2024 she was elected to the Fellowship of the Royal Society, and in the same year, even more unusually, was elected to the US Academy of Medicine, just indicating how groundbreaking and important her work is. So I'm really delighted to ask Sarah to now give the lecture.
Thank you. Thank you so much for inviting me to give this lecture. I'm incredibly honoured, and I'm really appreciative that so many of you came to listen. So one of the things that I was asked was to have a historical component because of the 50 years of funding fellowships and the 500th anniversary, and also I will, although my talk is partly aimed at scientists, I do have sections that are for non-scientists, so I hope that it's understandable, and I'm very happy to discuss anything afterwards. So I'm going to talk about genetic therapies. I'm going to focus on Huntington's disease, but I'm going to talk about several others that I think are really important in terms of therapeutics. And just to reiterate about from the Pewterers, we're incredibly grateful here that you chose the Institute of Neurology to fund fellowships, and with 50 years of funding, so many fellowships have been undertaken, and for that we're incredibly grateful to you, so thank you. So I wanted to begin, because I wanted to have some historical aspect, with a quote. So a quote from Aristotle, which is, "If you would understand anything, observe its beginning and its development." And so I came to Queen Square in February 1994, just from coming from an SHO job at the Hammersmith Hospital, and I had always been my dream, even as an undergraduate, to work for these two people. And so I came in February 1994, and I did work for Professor Anita Harding and Professor David Marsden, who would have an enormous impact on what I became interested in and what I became passionate about, which is neurogenetics, and in that, Huntington's disease, and in movement disorders. And so from these two giants, I would say that they set me on the path of where I am now. And so Huntington's disease is a really devastating inherited neurodegenerative disorder. It affects people in the prime of life, and we don't have currently any treatments. It's dominantly inherited, and it's caused by a CAG repeat expansion in the first exon of the Huntington gene. And it codes a protein, mutant Huntington, which exerts toxicity through multiple different Huntington protein species, which I will talk a little bit about. This is a lady from Lake Maracaibo in Venezuela, which was a really important group of people for the cloning of the gene. This is her age 38, and this is her 13 years later with end-stage disease. The mean age of onset of this disease is 42, so it affects young people. This is a lady, Kim, who is one of our patients in the Leonard Wolfson Center. She has these writhing movements, which are called chorea. She's got twisting movements as well, which are called dystonia, and she has evidence of dementia and neuropsychiatric symptoms. It also affects young people. This is a boy I looked after age 16. He has the more rigid Parkinson's-like picture. He sadly died when he was 21. His father died of Huntington's disease when he was 33, and Ammar's sister also died of Huntington's disease when she was 27. So you can see the effect this disease has on families because of its genetics. And this is what very severe chorea looks like. And we actually have quite a few patients like this. They end up having to nurse on the floor. This is an old video from the Movement Disorder Society, but it shows you what severe chorea is like. It is uncontrolled, writhing movements that are very distressing. So chorea, coming to the historical aspects, comes from the Greek word to dance. That's where the name originated. And the first descriptions of choreiform disorders was probably in the dancing plagues, which were named St. Vitus's dance in the 11th and 13th to 16th century through a multiple number of causes, but one of them is likely to have been Huntington's disease because it originated in England. And then Paracelsus in the 16th century, he was Swiss-born, he practiced all over Europe, In those days, people were multi-talented in the way that we aren't. He was a magician, a physician, a military surgeon, a medical reformer, and a philosopher. But like Aristotle's elements of fire, air, and water, and earth, Paracelsus's tria prima were mercury, salt, and sulfur. And his medicines were chemical combinations of these. And actually, if you look at the books written about Paracelsus, he actually killed quite a few people with these chemical combinations. But he was the first person to describe chorea naturalis. And this was the concept of chorea as part of a naturally occurring disease. And then Thomas Sydenham in 1631, who was an English physician, he was called the English Hippocrates, he accurately described the physical symptoms of chorea, and he described the physical symptoms of childhood chorea. He also studied rheumatic fever, but he didn't make that connection, and that connection was made later by Charcot. And then to George Huntington. In 1872, at the tender age of 21, he had just come out of medical school. He had spent a lot of his childhood and teenage years following his father around Long Island, where his father was a general practitioner, observing families in Long Island outside New York. He had one paper in his career, and just when he graduated, he described the cardinal features of Huntington's disease: the movement disorder, the chorea, the cognitive impairment or dementia, and the neuropsychiatric features. He presented his paper before the Meigs and Mason Academy of Medicine at Middleport, Ohio in 1872. And it was the only paper he ever wrote, but it's a beautifully written paper. And this is before anyone knew about Mendelian genetics. And this is the power of clinical observation. He wrote, "There are three marked peculiarities to this disease. One, its hereditary nature. Two, a tendency to insanity and suicide, and it's manifesting itself as a grave disease only in adult life." And in the same era as Mendel and his peas, and Darwin and evolution and his animals, Huntington and Bateson really applied genetics to humans. And this was from William Bateson around the time of the dawn of human genetics. William Bateson wrote, "The peculiar form of insanity known as Huntington's chorea very clearly follows the course of an ordinary dominant with few complications." And so this was very early and really understanding the genetic nature. This is the first recorded footage of someone with Huntington's disease from here at Queen Square. This is 1924. This is the gardens. This is a patient who has come up for a clinic appointment, very nicely dressed. He has early Huntington's disease. You can see he's got distal fidgety movements, which he's trying to conceal by putting them behind his back. His walking is a bit unsteady. When he turns, he's unsteady, and you can see he's a bit choreic. But this is early disease. Luckily now we don't take our patients into the garden square to video. But then genetics gave rise to eugenics. And eugenics was the scourge of the 20th century. So all the advances in genetics actually gave rise at that time to something bad, which was applied human genetics. And HD at this time was described as a curse from God, subjected to surveillance and controls, involuntary sterilizations. In the US, in the 1920s and 30s, you were forcibly sterilized if you had Huntington's in your family. It was a hidden secret in families. It was called the witchcraft disease, and it was even subject to murder in Nazi Germany alongside many others who were sent to the concentration camps. But many HD families were, as they were genetically unclean. And even in the kind of fairly modern day, in 1964, MacDonald Critchley, who actually was a predecessor of Mike Hanna, was the Institute director at the time in 1964. And this is what he wrote in an essay on Huntington's career. Relatives are liable to bear the marks of a grossly psychopathic taint, feeble-mindedness, insanity, suicide, criminal propensity, and drug addiction. Short of some drastic and unlikely eugenic intervention, the disease will not die out naturally. More likely, the numbers will increase from one generation to another like a hideous snowball. And that's from the director of the Institute of Neurology in 1964. So you can see the enormous stigma that patients and families with Huntington's disease have had over the centuries. In 1983, through a huge collaborative effort, the Huntington's disease gene was linked to chromosome 4. Now, it was the first locus to be mapped using restriction fragment length polymorphisms when the chromosomal location was unknown. So it was a really big effort. And this is Jim Gisella when he was very young, Milton Wexler, Nancy Wexler, and this is the paper. And then 10 years later, the gene was cloned. Now, this was a landmark paper in Cell. It involved 58 different researchers working all over the world, including Jill Bates, who you can see here. She was one of the people in group 2. And the project was led by Jim Gisella, Nancy Wexler, and the Hereditary Disease Foundation. It was a collaborative project across 58 co-workers. They had an agreement that the paper would be authored by the Huntington's Disease Collaborative Research Group rather than individual authors, so it was very fair. And lots of new techniques were developed, like chromosome jumping, to be able to clone this gene. So this was a massive effort. And the gene was found to be a CAG repeat in the first exon of the Huntington gene. And so now I'm going to switch slightly to give a kind of basic dogma of biology. Genes, DNA, make mRNA, which is your message, which make protein, which actually makes us who we are. In Huntington's disease, this is live cell imaging of brain neurons. This is over several weeks that have had live cell imaging. The Huntington is tagged, the aggregated protein is tagged with a green protein, and it's been speeded up, but the aggregation ends up killing the nerve cells. And that's what happens in vivo. And this is from a subject that we looked after in the TRACK-HD study. This is from 2008 to 2012, and you can see here there is already 16 years before onset the loss of their caudate nucleus, the ventricles are getting bigger, and they're losing structure around the cortex. And that is 16 years before predicted onset. So the disease is beginning decades before people show signs of the disease. And a lot of my work initially, and even now, was focused on lowering mutant Huntington protein. Mutant Huntington, this started in 2010. Mutant Huntington causes the disease. If we can get rid of it, it's game over. Not quite as simple. This is the gene, the message, and the protein. And the way that we first worked on and developed was a way of developing molecules that bound to the Huntington message, which means you switch off the Huntington protein production. And through a collaboration with Ionis Pharmaceuticals, that we took a molecule from scratch to the clinic. But the challenge of all of these quite exciting genetic therapies is how do you get them to the brain? And so this is a mouse brain, this is a rat brain, this is the largest rhesus monkey brain, and this is our brain. And we also have this blood-brain barrier which protects our brain, but it makes it very difficult to get molecules across. And so delivery and distribution of genetic therapies in particular to the human brain is currently our biggest challenge, because you can treat small areas, but to treat the whole brain, we currently don't have the tools with different types of genetic therapies. And so the work we first started on was an antisense oligonucleotide. This is DNA. And an antisense oligonucleotide is basically a single strand of chemically modified DNA that binds to its sister strand that is very specific. And so we started the work in 2010, started the trial in 2015. We spent a lot of work developing a really novel way of trying to get this drug to the brain. And there was a lot of work in that, with a fast push intrathecal bolus over 3 minutes and then get people to walk fast. And this was, at first, no one ever thought that this would work, that you would be able to do a lumbar puncture and get something to the brain. But actually, this was the result that everyone got excited about in December 2017, which was showing that you could get the drug to the brain, interact with the Huntington message, and lower the level of the protein. And this was December 2017. It made headlines all over the world. It was a really big news, because everyone thought, Huntington's is a genetic disorder. This has happened. It means that we're going to have treatments round the corner. And it didn't quite work like that. So the program was bought by Roche. It was a phase 1 study, and Roche took over the program. We're very excited by the phase 1 results, huge unmet need for this devastating disorder, and decided to invest in a massive phase 3. And they recruited nearly 800 people, gave them the highest dose possible of the drug, which was now called tominersen, the antisense oligonucleotide, and it was to be for 25 months. But then in March 2021, the trial was stopped, and it was stopped early from the Data Safety Monitoring Board. And it was stopped because the people in the active group were doing worse than the placebo. And this was the Data Safety Monitoring Group who saw the unblinded data. So they suggested stopping the trial, and importantly, stopping the trial. And it was stopped because of the toxicity of the high-dose ASO. And the maximum dose was chosen because of the delivery issue to get as much as you could into the brain with a lumbar intrathecal injection, and particularly into the deep structures of the brain. At the time, the trial design was really much about treating people late in the disease, and there were late-stage patients. ASOs have a risk of massive inflammation, and this was also lowering wild type. A post-hoc analysis suggested that if you were under 50 with earlier stage disease, you may benefit. And the post-hoc analysis resulted in a new trial called Generation HD2, which is ongoing, testing much lower doses. And recently, it underwent an interim analysis, and the results from the interim analysis suggested that it should continue. There were no safety concerns, but that everyone on this dose should move to this dose to improve the chances of showing clinical benefit based on the interim unblinded analysis from the Safety Monitoring Board. So we hope that this study will read out next year, and hopefully if the interim analysis continues to bear fruit, it might have a positive result. So these are all the different approaches that we're trying to develop in the lab: antisense oligonucleotides and interfering RNAs, zinc finger transcriptional repressors to switch off DNA, small molecules, and gene therapy. And this is a gene therapy trial we're doing with a microRNA against exon 1 Huntington, with an injection into the deep structures of the brain to lower the level of the protein. And this is the surgery. The surgery actually takes between 12 and 16 hours. It's very slow. This is speeded up, very much speeded up. And it's an injection into the chordatum putamen with 6 guides, and it takes a very long time to inject that volume, because the challenge about AAV is the potency of some of the AAV means that you have to inject. This was 1.2 mils, but it took 16 hours to infuse. So this trial was in very small numbers of people. and there was no really useful sham. So the regulator said that we could compare it to external controls that were from natural history data, which I think is an important step forward for rare disease. This is the low dose, this is the high dose, this is the external propensity score weighted matched control, and this is the rating scale of cognition, motor, thinking tests. And this score here, in the low dose and the high dose, even in numbers that are tiny, was statistically significant compared to the propensity score weighted matched control. So it looks like it's doing something. And this is CSF neurofilament. Many of you will know that this is a marker of nerve injury. And in the subjects, this is the natural history cohort. This is the pooled cohort. And there is an 11% drop at 24 months. These are very small numbers. I think the important thing is the regulators we're in discussion with the regulators, but the 36-month data is going to be the key data, but there is a buy-in for getting external control. So watch this space and let's see what the 36-month data looks like, and maybe this might be the first approved drug. So I'd like to just move slightly, because in the last 5 to 6 years, we've really now understand mechanisms of disease in Huntington's disease. And I think everyone would say to me, oh, the gene was cloned 30 years ago. Why haven't you got a treatment? And it's because the pathogenesis of the disease, and I think this is every brain disease, is much more complicated than we previously thought. It's not as simple as just lowering the protein. It's a slowly progressive disease as well, which means we have to do long trials. So I'm going to say the current understanding of where we are now, a lot of this has come from people the colleagues at the UCL HD Centre. Jill in particular has had a big influence on how we understand current disease mechanisms. And a lot of the work that we've done have contributed to this, alongside colleagues at MGH and Harvard and Rockefeller. And so what we think is happening now, and there's good evidence for this, is that there is a two-step pathogenesis to this disease. The first step is that you're born with the CAG repeat. Say you're born with 42 of the repeats in your Huntington gene. But during the lifetime, your lifetime, this gets bigger in the brain and it grows. And here, this is a southern blot, there are repeats in the striatum, the deep structure, of nearly 700. And some of the sequencing we've done have got repeats up into the several thousands, particularly in young onset individuals. So the CAG repeat grows during the brain. And the way it does that is DNA has to replicate. And because these repeats and these expansions are unstable, they cause these loop-out structures during DNA replication. And the DNA repair machinery tries to repair these loop-out structures, and it does it erroneously, and you get a bigger expansion. That's it simply. It's much more complicated than that, but that's how we think it's happening. And also, from a number of labs, genetic modifiers in DNA repair genes were all found to modify somatic expansion. And so this was linking cancer DNA repair to neurodegeneration. And what we know now is that growing of the CAG repeat that occurs in the brain is what drives the onset of the disease. But the toxicity is through a different mechanism. So that's step one. You have to have that expansion in the brain to be step one of the pathogenesis. So if we stop step one, we can stop potentially step two. But step two is the toxicity driver. And this is full-length Huntington gene. It's transcribed to the full-length Huntington message, but also it's transcribed with longer repeats. And in the lab, we've got a lot of data here now, and Jill's done a lot of work in this area as well, that once the repeat gets to about a 150 or 160, 20% of the Huntington that is produced, 20% of it is this Huntington 1A. And this causes the production of a very toxic exon 1 protein that Jill described and causes aggregation in the nucleus, which kills nerve cells. This full-length Huntington mRNA, the protein, undergoes cleavage and produces proteolytic fragments that also contribute to toxicity. So we know that we need to treat both of these as well as the step 1 somatic expansion to really make a big difference to this disease. So why is targeting somatic expansion so important? So you're born with 42 repeats. We know the repeat is getting bigger throughout the brain. Our work and Darren Monkton's work has shown that the more expansion you have, the earlier your onset. So if you have more expansion in the brain, you're just bad luck, and your DNA repair genes, the earlier the onset. Less expansion and slower onset. And many different DNA repair genes have been found. These are all well known in cancer, but these are all DNA repair genes that are known to modify Huntington's disease. And our work, and Davina Hensman, who was a PhD student at the time, identified MSH3 for the first time. And she found MSH3 to be a very powerful modifier of disease progression. And we showed that it was due to lower levels of this DNA repair protein. And if you happen to have lower levels of this DNA repair protein, you had much relatively slower progression. And the beauty of this, and this is where my thinking has changed, is this is a validated genetic target. So if you undertake drug development or validating targets, if Mother Nature, through human genetics, has already done the experiment for you, then you know that you have a better chance of success. So we know that people who have lower levels of MSH3 or another DNA repair gene, PMS1, or who have high levels of another DNA repair gene, FAN1, they all have slower disease. So Mother Nature's done the experiment, so they are very, very top candidates. And this is a very nice paper from the CSO of Vertex, that if you go for targets that have been validated through human genetics, you have an 80% increase in your chance of success for target validation. You just have to develop good drugs. So somatic expansion looks like it's really driving the whole disease. And this is a paper that we published recently. And this is showing that the somatic expansion rate in young people, decades before they're expected to show signs of the disease, predicts the earliest loss of nerve cells in the striatum with medium spiny neurons and the earliest increase in neurofilament, a marker of axonal injury. And so this tells us that somatic expansion 25 years before onset is driving the earliest signs of the disease. Now, there is a toxicity driver, but somatic expansion is the rate driver. It begins it. You need both. And so the useful thing about the paper as well as the science, and it tells you can do mechanistic science in humans, is the study showed that we have the potential to start planning prevention trials, because there's a treatment window when these youngsters, who are 25 years before onset, are all completely healthy. There is nothing to find. They're healthy on cognition, they're healthy on movement. But there are some very early changes, very subtle compared to matched controls, in brain volume, CSF biomarkers, and somatic expansion, that is now allowing us to have a suite of measures that allow us to do trials in stage 0 when it's decades before symptom onset. And that's really what we're working towards and raising money for, to do a prevention study in HD. And so just as an overview of therapies, there are many different approaches that we and others are developing. Genetic therapies, siRNAs, antisense oligonucleotides, zinc fingers, and so on. And for a rare disease, there's a lot of interest, and I think this is good. It's because there is a deep understanding of the biology now. There is a very good patient community group, a bit like cystic fibrosis and spinal muscular atrophy. Those patient community groups have been very powerful at getting drugs approved. And so we have a very powerful patient community group. And these are all the different approaches, looking at splicing, DNA and RNA, gene therapy, repeat expansion, oligonucleotides, regenerative medicine, inflammation. and many academic labs. All of us are working on these targets as well, and in red, the ones that are actually in trials. And so I think this has got relevance beyond just Huntington's disease, because one of the things that's coming out is that the mechanisms of repeat expansion diseases look similar for all the diseases. And so many of you will have heard of Fragile X, Friedreich's ataxia, Huntington's disease, spinocerebellar ataxia, myotonic dystrophy. And Arianna Tucci, one of our colleagues here, had a beautiful paper that many of us and the neurogenetics team were involved in Nature Medicine last year. And what Arianna's team did is they did whole genome sequencing of nearly 83,000 people worldwide. And she looked at the overall frequency of 16 repeat expansion diseases and found the incidence was 1 in 283. So much, much more common than previously thought. And actually the commonest was C9orf ALS, myotonic dystrophy, Kennedy's disease. And then Huntington's was 1 in 4,109. So rare, but not that rare. And all of these diseases exhibit that somatic expansion in affected tissue. The one disease that's been more difficult to study in this respect is C9 because the expansions are so huge, but even in disorders like Fuchs endothelial corneal dystrophy, they have expansions into the thousands, as in myotonic dystrophy. And what we and others have found that the same DNA repair genes modify these diseases as well. So there looks like there's a unifying mechanism for repeat expansion diseases. And if we can develop a therapy targeting MSH3, it'll be useful for all these drugs. And the other thing I think is super interesting, I find very interesting, every repeat expansion disorder, which means it's a disorder caused by a repeating sequence of DNA, every one that's been described all affect the brain. They're all neurological. So there's something about the brain and repeats that make the brain vulnerable. And I think that's a whole huge, big project in itself. But it's a very interesting finding. They're all neurological. So, moving on to other diseases. What can we learn from other diseases? And I've given you a whistle-stop tour of where we are in HD. There's lots going on. I hope we're going to have some therapies that are licensed soon. But look at some other diseases. And the next disease I'm going to talk about is not truly a brain disease, but it's a neuromuscular disease. And I think I see this as the poster child for genetic therapies. So spinal muscular atrophy is a disorder, and the common type is type 1, which affects babies. And if babies inherit the gene from both of their parents, they then have muscle weakness. By the age of 6 months of age, they're on a ventilator, and life expectancy is not much beyond 12 months of age. It's a terrible disease. And nusinersen was an antisense oligonucleotide developed by Ionis, and they developed a very cool way of switching in a missing part of the gene to make a functional protein to replace a missing protein. So the disorder is caused by missing key muscle proteins. And the way that the oligonucleotide was designed was to put back in the missing part of the gene, make a functional protein, and then deliver it. And Frank Bennett and Adrian Craner got the Breakthrough Prize for designing that oligo. But this was given as a slow infusion into the spinal cord to get as much as they could to the spinal motor neurons. This is the first paper in the Lancet in infants, and you can see here the data is so obvious. This was an open label, and you can see those that did not have treatment compared to those that had treatment, very clear difference. This was when, and the FDA then insisted on a sham for this disorder that children died of when they were 12 to 18 months, but the FDA were very strict about a sham. They are getting better now. But this was a sham, and this is the motor milestones respondents. And during the duration of this trial, everyone in the sham actually passed away. This was the interim analysis, and this is motor milestones. This was a halfway analysis, and this was the first time the trial was stopped after the interim analysis because it was unethical to continue the sham because the active group were doing so well. And this is in late onset spinal and muscular atrophy where it was also tested, but the effects are not as strong as in the babies. So my colleague and friend Richard Finkel in the US gave me a couple of these slides. He has now been responsible for setting up newborn screening for SMA in the US. So a little bit about newborn screening, because this is about genetics. 1963 was the Guthrie test, and then phenylketonuria, then hypothyroidism and cystic fibrosis. Then, in the 1990s, better ways of newborn screening were developed, and then in the 2000s, a DNA-based one was severe combined immunodeficiency disorder. And then, through Richard's work, in 2018, it was agreed that SMA should be added in the Uniform Neonate Newborn Screening Protocol. And it's now in 98% of infants born in the USA are screened for this, because it's not that uncommon. Globally, there's 9 countries in Europe, sadly not yet in the UK, Australia and South Korea. And I've had many conversations with Francesco Montoni about why don't we have newborn screening for SMA in this country? But there are challenges. But newborn screening is done in 98% of infants born in the US. And what that has allowed to happen is a prevention study. And so this is some videos from Richard. This is baby Chance. He was born with spinal muscular atrophy, and his older sibling died at 7 months of age because he was found to have SMA. So they screened the parents, then they screened the baby in utero. This next baby was also found to have SMA. And what Richard did was treat this baby at 9 days of age with nusinersen. Completely pre-symptomatic treatment, because the symptoms start coming about 4 to 6 weeks. And for many of you who've examined a baby, this is normal baby examination. So this baby is sitting, it has great head movement, it's active. If you saw when Richard picked it up, the baby has completely normal muscles. This baby has no signs of SMA. And this is the same child at 16 months, and he's walking, he's interacting, he looks slightly unhappy there, but you can see he's a normal baby. And this is actually one of his friends, and she's a normal child. And actually, when they're both examined together, there's nothing to find a difference. So this is, for the first time, a true prevention of a devastating disease. And I think although it's SMA, it's a neuromuscular disease, it's an infant disease, this is where we want to be for adult brain diseases, and it's very much where we want to be for Huntington's disease. Because I think if we treat early, I think we'll prevent the disease. And I think this is an example. These children are normal, whereas otherwise, as like the sibling, they would have died at 7 months of age. Just coming on to a few others for more common diseases like Alzheimer's disease. This is a tau ASO from Biogen, and Kath Mummery is running the study here at Queen Square, and it's a global study, and it's looking at an ASO targeting tau, and they were testing a number of different doses, but the thing I want to show you is this is tau PET, to look at tau accumulation in the brain, which contributes to Alzheimer's disease. This is 2 subjects, a baseline, who were recruited, but they were both on placebo. And this is them at week 25, and you can see here, accumulation of tau, no change. And then they were put on the antisense oligonucleotide, and at week 100, clearly tau clearance from the PET. So the drug is clearing tau. And actually, I met with colleagues in New York last week who had been at a meeting where Biogen was discussing this program, and it's moving forward, and the data is looking promising. And then also in Alzheimer's disease, and again, it's more common, is siRNAs targeting APP, and this is mivelsiran made by Alnylam. Again, these are slides courtesy of Kath, and this is a validated genetic target. John Hardy and colleagues found APP as a gene causing Alzheimer's disease, and this is a siRNA targeting APP. It reduces all APP products, and this is some of the early data. The ASO is given. There's very good lowering in CSF APP, and this is the placebo, and you can see the doses are once every 6 months, so it lasts a very long time. And there's marked reductions in the toxic components of Alzheimer's, A-beta 42 and A-beta 40, so it's looking promising. And it's safe, it was well tolerated, there was no evidence of inflammation, and it's moving forward. It's going to be in trials for Down syndrome and for cerebral amyloid angiopathy. And one day this might be used in the future when there's earlier Alzheimer's diagnostics for future prevention studies. So I think intrathecally delivered RNA therapies really came from our very early work, are now mainstream. Intrathecally delivered RNA therapies are being used for every disease. I've mentioned spinal muscular atrophy, SOD1 for ALS was approved, tau for Alzheimer's disease, alpha-synuclein for Parkinson's. You think of your target, design your oligo. That's the way. And it's not just ASOs, it's also siRNAs as well. And I think this is now a huge field of potential therapeutics. So many of you will have seen this baby. This baby was all over the news. You couldn't open any newspaper or any scientific journal. But this baby was the world's first baby treated with CRISPR gene editing. So whenever I do talk about Huntington's disease, someone always asks me about CRISPR. And I'm going to talk briefly about that. But this is an amazing story. I don't know if we'd be able to do this in this country, and this is why I want to have a call for arms. So this is a baby who was born and was found at 2 days postnatally to be failing to thrive. And the doctor did a number of blood tests, and they were found to have little boy was found to have sky-high levels of ammonia, which is toxic. And he was found to have an enzyme deficiency, which results in him making way too much ammonia. Now, ammonia is very toxic. And the only treatment for this is a liver transplant in the first 4 weeks of life. Now, to do a liver transplant in the first 4 weeks of life is very, very difficult, because it's very difficult to get donors. And plus, technically, livers at 3 weeks of life are tiny. So the team at the Pennsylvania Children's Hospital did whole genome sequencing on the child in the first month after diagnosis. They were still in hospital. They were getting sicker and sicker. If you don't get a liver transplant, then you pass away, usually about 2 or 3 months of age. This paper was published in the New England Journal of Medicine. But what I think is remarkable is this. This is the timeline. So the baby was born. Genetic diagnosis was within 2 weeks. They did whole genome sequencing and found there was a single base pair change in the gene that was meant to produce, it's actually an enzyme that is meant to control the levels of ammonia. It was a single base pair change, which allowed them to use a technique called base editing, which is where you change a single base. They screened for the most efficient and precise base editing approach in cells. They then made a mouse model in like 3 weeks. They then did off-target, which is very important. They met with the FDA and said, what do you think? The FDA was supportive. These timelines are tiny. A few weeks later, they had non-human primate toxicology completed. They then did the toxicology in mice because they needed to batch test. They made a clinical batch of the CRISPR gene editing. They tested the batch. They did off-target. They then went to the FDA, who granted them approval in 5 days to do the drug, to release the drug. And this was a collaboration between CHOP, the Children's Hospital, and Rebecca Arends, who I know well. She's a gene therapy doctor and a pediatrician. Danaher and Fyodor Unov, and Berkeley, where Jennifer Doudna is. It was also with colleagues at Harvard, David Liu and Ben Kleinsteiver, who really led the work on the base gene editing. It was a massive number of people who came together to work fast. And the FDA were amazing. This is pre-Trump FDA. So it might be different. But we just can't get anywhere near this in this country, and we can't get anywhere near this in Europe. But this is how responsive we've got to be. We need an MHRA that can do this and give us an approval. And the IND application was submitted 2 weeks, it was actually 5 days later. It was approved. The baby got the first dose because the baby was really failing by then. And they received the second dose, and the baby is so much better. So CRISPR has enormous potential for eye and blood and enzyme and gene replacement. For adult brain diseases like Huntington's disease, CRISPR is decades off because we can cut out the CAG quite easily in neurons, but it's how you do that and deliver the machinery to the entire human brain. And that's the challenge. I think CRISPR for small regions in the brain will be possible. And I think it will be possible for pediatrics, for children with very small brains, because the delivery is easier. But for adult human brains, I really think we're far away from genome editing the whole brain, because you just won't get enough into all of the different neurons that need it. But this, for me, this is amazing. So I think for us, in Huntington's disease, we really want to be where they have been in SMA, and we want to be able to do trials decades before onset, where we know so much about this disease. We know what begins, we know what it's caused by, and if we can set up the infrastructure to do a prevention trial, that's exactly what we're trying to do at the moment. And so I hope that if we treat people when they're 25 years old, that we prevent Huntington's disease ever occurring. And I think that's what we want, that's what they've done in SMA. And where is all the work happening? The work is all happening here in many different diseases: Alzheimer's, Parkinson's, ALS, here at the Institute of Neurology and at the National Hospital for Neurology. And I hope that we will have the same successes as SMA and also the baby with the CRISPR gene editing if we can get our regulators to be very responsive. And so my vision is a world in which families don't have to worry about Huntington's disease anymore, don't have to feel ashamed, and don't have to feel stigmatized. And that's what we're working towards. And this is the final quote from Martin Luther King. We must accept finite disappointment. And I think if you're trying to develop therapies for these difficult diseases, you have to accept disappointment. But what's important is we must never lose infinite hope. So thank you for listening. I'd like to thank everyone who works with me, my lab team, the HD Young Adult team, my trials team, the HD Centre, Ed and Jill and Rachel, who are the co-directors, and thank you for listening.
So thank you, Sarah. What a tour de force, not just history, not just fabulous science, but unlike your health warning, even to a pig ignorant lawyer, it was all comprehensible, which is really quite an achievement. And it struck me as you were telling that long historic story, the importance of investment in the spirit of hope. Yeah. And of course, the story of the Pewterers Company, the story of so many of the livery guilds, it's about investing for the long term and investing for the long term in research and education for the remarkable dividends that it can pay. And we've seen in so many of these diseases remarkable progress that builds upon a history of not only financial but also the kind of intellectual investment that a place like UCL describes. And so, Sarah, very much your tour de force describes a process that we deeply believe in. It's a process of investment for hope in change. And we know too that that's a process that happens really well in universities. Because the other thing that became clear as you were talking was that this was a dialogue not just between basic scientists and clinicians, or clinicians and families and communities, but also a dialogue between clinicians and philosophers, as we thought, for example, about the history of genetics and the difference between genetics and eugenics, fine as it sometimes might be, but also the importance too of social scientists, as we think about mechanisms for approval, mechanisms for drug discovery. The CRISPR story about the overproduction of ammonia in all sorts of ways is a triumph, but it's equally possible that in 50 years people will be saying, you know, they got excited at that particular moment and then they foreshortened the whole approval processes and all sorts of disastrous things happen. You need social scientists, you need philosophers to help think through how you realise the clinical benefits of this remarkable treatment, these remarkable treatments. Investment in the future in hope, investment in the future in hope in partnership with people working across disciplines to deliver cures to devastating diseases. That's a kind of miracle that you see and that I see every day in this remarkable place, this centre of the globe, as we now know from the final slide, at the hands of people of this kind of calibre here in the Institute and right across the university. And so please join with me again to thank the Pewterers, to thank Sarah for a remarkable lecture, and to congratulate all of you across brain sciences at UCL for the extraordinary work that you do. Thank you.